And if we break apart the acronym, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. Now that's a mouthful, but it does tell you the two main parts found in CRISPR. First of all, we have the repeats. These are going to be short segments of DNA, so 20 to 40 letters in length, and they're going to be palindromes. Remember, a palindrome is a sequence of letters that read the same left to right, like never, odd, or even. So we're going to have these letters that are palindrome. The reason why is that when you transcribe that DNA, you make RNA that forms these little hairpin turns. So we've got the repeats. Those are all identical.
CRISPR is a technology for changing the sequence of DNA in cells in a precise fashion to correct mutations that might otherwise cause disease.
The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together.
It's a very exciting technology that's going to do a lot of good in human society and for human health. It stretches from human therapeutics to agricultural applications to thinking about how do we make better biofuels. These would be incredible outcomes, I think, of using this kind of technology in the future.
Regardless of your personal take on genetic engineering, it will affect you. Modified humans could alter the genome of our entire species because their engineered traits will be passed on to their children and could spread over generations, slowly modifying the whole gene pool of humanity. It will start slowly. The first designer babies will not be overly designed. It's most likely that they will be created to eliminate a deadly genetic disease running in a family. As the technology progresses and gets more refined, more and more people may argue that not using genetic modification is unethical because it condemns children to preventable suffering and death and denies them the cure. But as soon as the first engineered kid is born, a door is opened that can't be closed anymore.